Each year Wageningen University Fund awards a prize. This year's prize is for outstanding research. And I'm very happy to see that also this year there was a lot of fascinating research to choose from. Papers in Nature, papers in Science, papers in Brouns, all kind of really, really good science. But only one researcher can win the prize, of course. Karel Mak, President of Wageningen University Fund, may I invite you to come over to award the prize.
Mr. Rector, Mr. Mayor, distinguished professors, ladies and gentlemen. As most of you know, the Wageningen University Funds seeks to promote the success and reputation of this university by taking and supporting initiatives aimed at adding value to the university and at stimulating excellent education and research. As the Rector said, one of these activities is the awarding, once every 4 years, of the Research Award on the occasion of this Dies Natalis. The first time it was awarded was in 1959, so we did build a certain tradition here, and a valuable one too. The prize is awarded to a young researcher at the university to recognize his or her excellent scientific publication written in the 4 preceding years. It is good to note that the prize appears to have acquired a certain predictive value in the sense that many winners have gone on to become a professor at Wageningen University. The prize consists of a formal certificate, a judicium, and a monetary award to be spent to the winner's own liking. Ladies and gentlemen, the jury for this year's research award has the following members: Professor Martin Kopp, Rector Magnificus and President of the jury; Ton Bisseling, Professor of Molecular Biology; Rick Leemans, Professor of Environmental Systems Analysis; and Arthur Mol, Professor of Environmental Policy. I would like to thank these 4 jury members for their work, which has resulted in a unanimous judgment. The jury has awarded the Wageningen University Research Award to the following publication in Science: Small CRISPR RNAs Guide Antiviral Defense in Prokaryotes, written by Stan J. Brouns under the guidance of Professor Johan van der Oost. It is a very appropriate choice in view of today's theme, Science for New Frontiers. And I will briefly explain to you why by describing the subject of this publication. What is it that Stan Brouns has discovered and described? He has found that the microbial genome has a region in which we can find specific repetitive sequences, and that in between these sequences, genetic sequences of a viral nature appear to be interspersed. RNA strings generated from these areas appear to contain these viral sequences. This is intriguing indeed, but in itself it is not a huge shock, because such an RNA interference mechanism was already known from eukaryotes, higher forms of life. This discovery has been awarded with a Nobel Prize, and David Bolkamp has received an honorary doctorate for his research in this area at this university. However, Stan Brouns did more. He identified the enzyme that carves out the small RNA string containing the viral sequence, and, most importantly, he showed that this is used as a defense against a virus that attacks the microbe. So, microbes build an immune system by integrating small fragments of viral genomes in a specific place in their own genome. These specific places are called CRISPs. And these specific locations in the microbial genome serve as a memory of previous viral attacks, like our own eukaryotic immune systems do. But this microbial memory is part of its DNA, so it is also transferred to daughter cells. Thus, Stan Brouns has, together with his research team, discovered a major immune system that bacteria use to survive viral attacks. It is a system of stunning natural beauty. The prokaryotic immune system appears to be an extremely sophisticated defense technology. It is not only beautiful, now that it has been unraveled, it also appears to have practical applications, as the patent award clearly indicates. Thus, this paper has become a cornerstone document of CRISP's research that has greatly expanded internationally since its publication. It has proven to be the fundament of a major new research line, not only in science, but also in other prestigious journals like PNAS, Nature and Cell. Articles about this research have been published. Just a few words on the history of this work. This piece of research was started only 2 years before this publication was made. In 2005, a major grant came available to the research group of Professor Johan van der Oost. It was a prestigious Vicky grant of 1.2 million euros awarded by NWO. With this, Stan Brouns was recruited as a postdoc. Upon hearing rumors about a hypothetical new defense system in microorganisms, Stan and John decided to focus their attention onto CRISPs. They evidently had a good grasp of which uncharted terrain would have major discoveries in waiting. They also chose the right organism to work with, E. coli, that appeared to be a perfect model system and thus helped them to stay ahead of the competition, like a major research group in Lithuania, I just heard. It is the combination that Stan displayed of a great sense for developing novel ways of thinking in biological research and experimental skills of great excellence that led to this seminal publication. This discovery has caused biology and biochemistry textbooks to be rewritten and extended with an entirely new chapter. And it has opened new ways of efficiently protecting microorganisms in biotechnology. Last but not least, I should note that Stan Brouns has played an important guiding role in a team of enthusiastic, talented young scientists at this university. Stan Brouns, may I invite you to accept the Wageningen University Fund Research.